Hello. Now in this module, we'll be looking at the Avery, MacLeod and McCarty's experiment, which went on to prove that DNA was the transforming principle. So let us see what is this experiment. Now in 1928, Griffith tried to prove that DNA was the genetic material, and 16 years after Griffith's experiment, which means in 1944, three scientists, namely Oswald Avery, Colin MacLeod and Magdalen McCarty, tried to conduct an experiment once again using the same bacteria called Diplococcus pneumoniae, a bacteria that causes pneumonia in mammals. Only this time they did not use any mouse for the experiment. They conducted their experiments only in the test tubes. Let us see what did they do in their experiment. Now what they basically did was they took the heat-killed S-type and tried to isolate and purify the components of the cell. Okay? What did they purify and isolate? Different components of a cell. Now let's recollect the different components of a cell. They would include the DNA, they would include the RNA, they would include proteins, they would include lipids, and they would also include sugars or carbohydrates. These were the several different components of the heat-killed S-type cells. And they individually mixed each of the component with the live R-type to see which one could transform the live R-type into the live S-type. Okay, so let us see what did they do and what were their conclusions ahead. Now what you see here are basically 5 different set of test tubes which basically contain the live R-type bacteria. Now what they did was basically individually mixed each component from the dead or heat-killed S-type and mixed it with the R-type. Fine. So what we are doing next basically is taking different components from the heat-killed S-type bacteria. When proteins were taken and mixed with the R-type cells, no transformation was seen and the R-type cells remained R-type. When RNA was taken from the heat-killed S-type and mixed with the R-type cells, once again there was no transformation and the R-type remained R-type. Then when lipids were taken from the heat-killed S-type and mixed with the R-type bacteria, once again there was no change and the bacteria remained R-type. And then when sugars were taken or carbohydrates were taken and mixed with the R-type bacteria, once again there was no transformation and it remained R-type again, as you can see in these 4 pictures here. Okay? Finally, when DNA was taken from the heat-killed S-type and mixed with the R-type bacteria, they got transformed into S-type. Is that okay? So what happened here was only those ones which were mixed with DNA were transformed into S-type bacteria. Is that okay? To confirm this finding, they did a reverse experiment using certain enzymes. Now what they did here was took a mixture of dead S and live R-type bacteria. This mixture was treated with enzyme protease. And what observed was live S colonies were formed. Why? Because remember, proteases are enzymes that will digest protein and not the DNA. Since DNA was still alive, transformation happened and the live R turned into live S. In the next set, what they did was they took a mixture of dead S and live R and treated this mixture with RNases or ribonucleases. Once again, they saw that it changed to live S type. Why? Because ribonucleases or RNases will digest the RNA and not the DNA, which means transformation will again occur and the live R will change to live S. In the last case, what they did was took a mixture of dead S and live R and they mixed it with deoxyribonucleases or DNases, and they saw there was only live R cells found. So live R and live R, there was no change. Why? Because DNases digest and destroy the DNA. So when DNA is destroyed, no transformation will happen, which goes on to prove that DNA is the transforming principle. Is that okay? So let us see what were the conclusions of Avery, MacLeod and McCarty's experiment. They concluded that protein digesting enzymes or proteases did not affect the transformation. Then RNA digesting enzymes or RNases also did not affect transformation. But only when DNA digesting enzymes or DNases were mixed or added, they did inhibit transformation, which goes on to prove that DNA is the transforming principle. Even after they proved that DNA is a transforming principle, the other scientists were not believing that DNA is the genetic material, okay, till another set of experiments are conducted. Okay, now for that we will be looking at the next module. So what you saw in this module we only saw the Avery, MacLeod and McCarty's experiment, which proved that DNA was the transforming principle. Okay, the next module we'll be looking basically at the Hershey and Chase's experiment. Okay, thank you and take care.